Hey bio students, this is Mr. Wolf, and today I'm going to talk to you about the animal terms you're going to need to know for the animal unit. So to first start the animal terms, we'll start talking about animal symmetry. Now animals have very interesting symmetrical patterns. There aren't many animals that follow the asymmetrical body plan. Pretty much the only asymmetrical animal that you're going to find where there's no body symmetry at all are the sponges. That basically means there's no rhyme or reason or shape to the way the organism is put together. A lot of animals show radial symmetry. Basically, you have similar parts that branch out in all directions from some sort of central line. The best example of radial symmetry, if you think of like a sand dollar, if you've ever seen a sand dollar before, you have this radial symmetrical pattern that forms, where basically with a radial symmetrical organism, you can think of it that you can cut them in any direction and you would have the same or two equal halves. What radial symmetry gives them, it gives them a top and a bottom side, but really no front or back side like you think of us. Most cnidarians are radial symmetrical. There are some other types of organisms that, like starfish, that show what's called pentaradial symmetry. Now, another example of symmetry is found in organisms like us, which is called bilateral, which the bi part of this means two. Basically, if you look at a human being, and we'll draw a nice little stick figure here, you can cut a human, if you were to divide them in half, straight down a midline here, which would give you two equal halves. Now, internally it might not be equal, but externally it's pretty equal if you were to cut right down the middle of our nose. What this gives us is several different sections of our body. We have what's called a dorsal and a ventral side, the dorsal being our back, ventral being our front, like where your stomach is. You have an anterior and posterior. Anterior would be where your head's. Just think of antlers on a deer. Posterior would be the opposite of that, like where you're sitting. And then you have a left side and a right side of your body. That would be all characteristic of a bilateral symmetrical organism. Organisms that show bilateral symmetry also exhibit what's called cephalization. We'll talk more about that later, but it's a concentration of sensory organs and a brain near the anterior or the head region of the organism. Now that we've talked about symmetry, we're going to talk about body cavity. Body cavity, if you think of it, it's where all your organs fit inside of you. So we have several different kinds of body cavities, but we first have to learn that basically the body cavity is based on the embryological tissues that develop as we're developing before we're actually born. And we actually develop three different tissue layers. We have what's called ectoderm, which is kind of like the outside tissue. This would be like your skin. You have mesoderm, which is like the middle tissue layer. That'd be like your muscles. And you have endoderm, which is the most inner tissue, which is your digestive tract. So there are some organisms that don't have a body cavity at all. These are called acoelomates or acoelomates. I've heard it pronounced both ways. But this is where all three body cavities are kind of sandwiched together. You can see in this diagram of a flatworm. You see right here we have the endoderm, that's the digestive tract, sandwiched right up to the mesoderm, that's all this red tissue. That'd be like the muscles that move the flatworm. And then we have the ectoderm here, which is the outer skin. There is no actual cavity between any of those layers. We also have what's called a pseudocoelomate, where we have this prefix pseudo, kind of means false. This is like a false body cavity. It's not a true body cavity because the digestive tract, this inner endoderm layer, is not lined with the mesoderm muscle layer as pictured here. You can see there is a cavity right in here. Let me switch to red. There is a cavity in here, but it's not lined by mesoderm completely, so we call these false cavities or pseudocoelomates. The example that's pictured here is a roundworm. And then finally we have coelomates, or again I've heard it pronounced coelomates, is where you have a cavity fully lined by mesoderm. So there's our digestive tract right there, and then this is the mesoderm that fully lines the digestive tract. So you can see right here we have a cavity, and here we have a cavity too, and that would be considered our coelom. So the mesoderm lines and supports the gut, the digestive tract, and the internal organs are kind of suspended in the coelom. If you think of our body, we have two coeloms. We have a coelom right here where we have our heart and our lungs and other organs, and then our diaphragm separates that into a lower coelom where our stomach and our digestive organs all fit. So we are considered coelomates. Next, we're going to talk about nutrition and digestion. Now, basically, most animals get their nutrition by ingestion. We bring food into our body. We're heterotrophic. We have to eat other organisms for food. We bring it inside and digest it. What the major difference between a lot of animals is if they're one or two openings. Obviously, we have two openings, but there are some organisms that have what's called a gastrovascular cavity, where it's a single opening for digestion and ingestion, and then there's also the same opening for getting rid of waste. So basically food comes in that opening, it sits in a little cavity, it gets digested by digestive enzymes, and then anything that's not digested leaves out that same cavity. We see this in cnidarians, ctenophores, and flatworms. All are organisms with only one opening. They're the gastrovascular cavity. Now, organisms like us, we have an alimentary canal where we have two openings for digestion. Basically, this allows us to continually eat. We don't have to wait for our small intestine to be empty for us to fill our stomach up with food. Since food is constantly traveling in one direction, this allows us to eat more and basically get more energy and be more active. Whereas cnidarians and flatworms, once they fill up their gastrovascular cavity, they have to wait to get rid of the waste before they can fill it up again. This is a big evolutionary jump for us, for animals, that allowed us to basically eat kind of on the run. We've also developed many digestive structures that aid in producing enzymes and storage units and so forth for the food that we eat. Now we're going to talk about circulation. How do we get the energy that we have, or how do we get nutrients around our bodies? The first example is just by simple diffusion. Now, all organisms use diffusion, which again is the movement of molecules from a high to a low concentration, because it requires no energy. And remember, osmosis is just a diffusion of water across a membrane. Now, all organisms use diffusion in some way, but other organisms, as other animals, as we've gotten bigger, have to have certain systems to move things around their entire body because the cells that need those nutrients aren't close enough to the environment for simple diffusion or osmosis to work. So some organisms have developed an open system for circulation, where basically you have circulating fluid that just kind of fills the organism's body. There's no vessels that contain that fluid. So the organs are just kind of bathed in this nutritious fluid, and it just kind of moves nutrients and oxygen and so forth around the body. This is found in insects and crayfish and some mollusks and other invertebrates. Basically, smaller animals tend to have open systems. We have a closed system where we have a continuous series of vessels. These are our veins and our arteries and capillaries that are all different sizes that deliver some kind of blood or nutrient-containing fluid to the body cells that need it, and it's usually pumped by a heart, so therefore it has to return to the heart to get basically filled back up with nutrients and then pumped back out. These are found in echinoderms, and vertebrates all have a closed system. Now, when talking about closed systems, we've got to think about how many different loops does the circulating fluid take. For example, in humans, we have 2 loops, but in fish, there's only 1 loop that the nutritious fluid takes from going from the heart to the body cells back to the heart. Speaking of the heart, animals have different heart structures. Animals that do have a heart have the different number of chambers in the vertebrate group of animals. Now, fish, for example, have 2 chambers. You have an atrium and you have a ventricle. Basically, the atrium receives blood from the body, it pumps it to the ventricle, and then the ventricle pumps it out to the gills, and then it makes a single loop back to the atrium where it started. So it's 1 loop, 2-chambered heart. Not very efficient, but it works well enough for organisms like fish. Amphibians and reptiles have a 3-chambered heart. In fact, there's one group of reptiles that has a 4-chambered heart, but I'll let you guys figure out which one that is. Basically, you have 2 atria that sit on top of 1 ventricle, and the thing about the 3-chambered heart is that it's pretty efficient, but you get a mixing of oxygenated and deoxygenated blood in this single ventricle. The 4-chambered heart is probably the most advanced heart, and this is found in birds and mammals. And again, there's one reptile that does have a 3-chambered, or there's one reptile that has a 4-chambered heart. But this basically we have our 2 atria that receive blood, and then 2 ventricles. Again, it's not a very good drawing, but 2 ventricles that pump blood back out to the body. So let's say we start here and we pump blood to our lungs to pick up oxygen. This blood comes back to an atrium, gets pumped down into a ventricle, which then pumps that out back to the rest of the body. Then the blood comes back to the atria, goes down in the ventricle, and makes its second trip. So you can definitely see there is 1 loop here and 1 loop here. This is why we are a 2-looped system for circulating fluids and nutrients around our bodies. Speaking of circulation, we're going to start talking about respiration. This is basically how we get oxygen and get rid of carbon dioxide. Now, a lot of organisms that have their cells close enough to the surface of their skin or their environment use simple diffusion. There's no special system for them to get oxygen in and get carbon dioxide out. Other organisms, bigger animals, need certain structures to help this gas exchange. For example, aquatic organisms usually use gills. They're specialized structures that exchange gases in water. Organisms that live on land use some kind of lungs or some kind of tubes to transport oxygen and gases in and out of their bodies. For example, spiders, arachnids, use these things called book lungs. It's basically small membranous folds that look like the pages of a book. Insects use these things called tracheal tubes. So it's basically, if you look at the insect, I'll draw a very crude ant here. They have these little tiny tubes that allow gas to get into the ant's body tissues and get to the cells that need that gas, especially oxygen. Organisms like us use lungs. We have, it's a respiratory organ where gas exchange occurs. Invertebrates, we have these tubes that connect our pharynx, which is basically the back of your mouth or your throat, to those lungs. Those are called trachea tubes. And they branch off into bronchi and bronchial tubes and get smaller and smaller and smaller until you get these little alveoli in your lungs where the gas exchange actually takes place between these little tiny air pockets and the blood vessels or the capillaries found in your lungs. But it's interesting, at the very smallest level, we still rely on simple diffusion for gas exchange because what's nice about diffusion is that it requires no energy to exchange that gas. Once the oxygen gets into our lungs, it takes no energy on our part to get the oxygen into our blood. We use simple diffusion as a mechanism for transporting oxygen in and transporting carbon dioxide out. Now moving on to reproduction. When talking about reproduction, we got to look at basically where does reproduction take place, and then what kind of birth does the organism have. So where it takes place, we can either have external, which is outside the body, and internal, which would be inside the body. Now in all animals, the female, if they are separate sexes, produces the eggs, and the males produce the sperm, but you'll find that there are some animals that actually have both reproductive organs, and they can't fertilize themselves, but they can be both the mommy and the daddy, depending on if they're fertilized or if they're their fertilizer. But basically, if it happens externally, that means the union of gametes, so sperm and egg, unite outside the bodies of the parents. This would be examples would be like fish and amphibians like frogs, where the female lays the eggs and then the male deposits the sperm outside the body. Internal fertilization, which kind of ups the chances of the sperm finding the egg because it's in an enclosed environment, takes place inside the female's body. So there has to be some depositing of sperm cells inside the female for the sperm to find the egg. The female never releases the egg into the environment. These are found in reptiles and mammals, and some cartilaginous fish have internal fertilization. These would be like sharks, for example. Now, organisms that lay eggs can either lay their eggs in water, or they can lay their eggs outside of water, or basically keep their eggs inside the animal itself. So basically, an amniotic egg is just an egg where the embryo is encased in a self-contained aquatic environment. So the organism is not actually released into the environment until it's developed further. Amniotes are organisms that produce what's called an amnion. These are like your reptiles and birds and even mammals. Even though mammals don't lay eggs, except for there are 3 examples that do, we basically keep our developing embryo in this self-contained environment called the amnion, which we get the word amniotic fluid from, until the organism is ready to be born. Now, speaking of types of birth, we're going to talk about 3 different ways organisms are born. We have what's called oviparous, where the eggs hatch outside the mother's body. For example, most reptiles, all birds, and there are actually 3 species of mammals that actually lay eggs. These are usually calcified, hard eggs that have to hatch. Usually the mother either incubates them or leaves them in some kind of warm environment that eventually they hatch, and those animals are kind of on their own. Then you have what's called oviparous, which is an interesting word to say, where the eggs are actually stored inside the mother right before birth happens, or they're actually born kind of alive, but there still are eggs that form. Good example of oviparous animals would be like sharks. Certain kinds of sharks kind of give birth to live young, though it's not the type of live young or live birth that you think of when you think of mammals. And the last type of birth is called viviparous. Viviparous animals form no shell around their eggs. The young are kept inside the mother's body until they're mature enough to be born. Most mammals, except for the 3 that were mentioned up here, are viviparous, but there are a few species of lizards and snakes that are also viviparous. When looking at animal development from that single cell, which we call the zygote, to a multicellular organism, there's two major pathways that animals follow. Basically, when you go from that 8-stage cell, where you basically are just a ball of cells, to what's called the early embryonic stage, we'll call it, where you start developing this archenteron, which is basically an early digestive cavity. This is where you see the major difference. Protostomia, which is comes from the word mouth first. Basically, this first opening right here, this blastopore, where you get that first opening and you get this cavity that develops, will eventually become the digestive tract, is what develops into the mouth. What's interesting about this type of development is that during this 8-cell stage over here, you get what's called spiral and determinate cleavage, which does not allow this mass of cells to divide and result in identical twins. Deuterostomia, which the blastopore, if you look down here, this blastopore turns into the opposite end of our digestive tract, the anus. The mouth develops later. What's interesting about this type of development is that you get radial cleavage in that 8-cell stage, and that radial cleavage allows this cell mass, if it separates, it can separate cleanly into two masses of cells that are equal, and this allows for identical twins. So the reason why humans can develop identical twins is because we're deuterostromia in the way we develop. Protostomes don't develop in that same way, and you can't have that identical twin characteristic. Now next we're going to talk about water regulation and excretion. These kind of go hand in hand with each other. Basically, when talking about water regulation, we have to worry about how do we remove excess water. For example, animals that live in freshwater environments have to get rid of excess water that might be filling up their cells. Animals that live in a very hypertonic environment or live on land have to worry about conserving water in their bodies because water is not always available. Closely tied to the water regulation is the excretory system. So there are some terms and structures we're going to talk about. Basically, excretory system is in charge of removal of nitrogenous waste, which is basically in the form of ammonia, which comes from the breakdown of proteins. So as you digest proteins, that nitrogen forms ammonia, and that's very toxic to your body. A lot of animals, since they live in aquatic environments, don't really have a problem getting rid of ammonia because they just kind of excrete it out of their body and just floats out in the water. But there are other animals that live on land that have to have some kind of system to get rid of this waste product. In flatworms, we see these things called flame cells, which basically collect excess body water and transfer it to the excretory system. In insects, we see these things called Malpighian tubules, where they collect bodily fluids and they basically dump them into the intestines. The water is reabsorbed by the body because they're mostly terrestrial animals, and then the waste leaves the body as nearly solid. In other animals, like earthworms, we have these things called nephridia or metanephridia, which are basically little tubules that eliminate cellular waste and excess water. Some water is reabsorbed back into the blood vessels. And then in bigger organisms or bigger animals like us, we have things called kidneys, which are basically big collections of these types of tubules all in one place that act as filters. They filter out waste, the nitrogen compounds from our blood, while helping regulate water levels in our body. If there's too much water, we'll get rid of it. If there's not enough water, our kidneys will conserve it. Next, we're going to talk about the support system, basically what gives our body shape. There are three basic different types of support systems found in animals. We have what's called a hydrostatic skeleton, where some kind of fluid-filled cavity, where you get the word hydro from, is surrounded by muscles, and the pressure of that fluid, so the muscles basically push on the fluid to get the animals to move. These are examples are like echinoderms, cnidarians, annelids like earthworms, nematodes have a hydrostatic skeleton. Basically, soft-bodied animals, where if you were to step on them, don't really go crunch, they go squish, would be your hydrostatic skeleton type animals. Exoskeletons, which are hard external coverings, found in some invertebrates, provide both protection and support. These are like in your arthropods, like insects, spiders, and crustaceans. And then shelled mollusks have exoskeletons. I always like to think of a lobster as a good example of an exoskeleton. What's interesting about organisms with exoskeletons, they have to shed those exoskeletons in order to grow. So they actually have to molt. I remember one time when I was a kid, my dad brought home the exoskeleton of a tarantula. It looked just like the spider. It looked like the tarantula, but it was just its outer covering. The tarantula had molted. It had gotten rid of it in order to get bigger. Now, the advantage of what we have in endoskeleton is that we don't have to shed our skeleton to grow. Our skeleton's found inside our bodies, and our muscles pull on those skeletal bones, for example, and allow us to move. Chordates, which are what we fall and have exoskeletons. Echinoderms also have endoskeletons like starfish. And then porifera, the sponges, the very simplest of all animals, even have an internal skeleton, not made out of bone, but made out of other compounds that give them some internal structure. Finally, we're going to talk about the nervous system. Basically, in the nervous system, we want to look at how does the body communicate to each other and how do we respond to our environment. So many animals have developed these structural cells. Many animals have developed these cells that act as nerves. They're specialized cells that transmit electrical signals throughout the body. It's a way of communicating. Now, when these nerves start forming these little clusters, we call them a ganglion. It's basically kind of a collection of nerves that help kind of centralize where signals are going. So we see these in very primitive animals. Some other animals have these things called nerve nets, which are like web-like systems of neurons. These are characteristics of radial symmetrical animals such as sea stars. Now, organisms like us, the more complex organisms, have what's called cephalization. We mentioned this before. It's a concentration of sensory equipment at the anterior, in this case our head, part of the body. So this would be an uncephalized person, and this would be a cephalized person. You think of cephalization, you think of all of our senses, our sense of sight, our sense of smell, our sense of taste, our sense of hearing, are all closely concentrated at one end, which is also very close to our brain, which is our organ. That's basically a big collection of nerves. It's a central nervous system main structure that processes and integrates information that is collected by the periphery nervous system, which basically comes from all the body parts that we have and all the sensory organs we have. So we are cephalized. Most animals that you probably think of, even the very small ones, are cephalized. Now this diagram serves as an overview of the major animal groups and some of the terms we had just talked about. For example, you can see here, we see different types of symmetry. Here's the bilateral symmetrical group, so everything above this point right here, so all the way from Platymenthes to chordates, like where we fit in, are bilateral symmetrical. These two are the radial symmetrical organisms, cnidarians and centaurs. And then there's only one group that's not symmetrical, that's porifera. Those are your sponges. You also see within the bilateral symmetry we have coelomates, and we have pseudocoelomates, and we have the acoelomate group. Only one group is acoelomates, that's Platymenthes, the flatworms. Pseudocoelomates, we have two, and then everything else, actually no, we have three. Nematodes are also pseudocoelomates, and then everything else from here, from mollusks to annelids to echinoderms and organisms like us, chordates, are all coelomates. And within the coelomate group, we see protostomia and we see deuterostomia. We're not going to talk about this group here. We're not going to worry about them. Basically, we see a lot of the terms that we've talked about, and you can see how we move up this phylogenetic tree. We see more and more complex animals as we move from left to right. Now, this is a lot of information I just talked about, so I highly recommend rewatching this video and making sure you add some good notes to the sheet we handed out to you. I hope this was helpful, and I'll see you in class.